Okay. So we are going to start disaccharide. From the name, it can be explained that what is disaccharide. Di means two and saccharide means sugar. So disaccharides means when we hydrolyze this molecule, it will produce two molecules of monosaccharides. And if we want to hydrolyze monosaccharide, then it will lose its characteristics for carbohydrates. So the monomeric units are monosaccharides, and when they are joined together, they are called disaccharides. Okay, so you can see the examples. This is lactose, this is maltose, this is sucrose, this is trehalose. All of these are disaccharides. So you can see that lactose here, this one is a glucose molecule and this one is a galactose molecule. So glucose and galactose are joined together via a bond, and this is called the glycosidic bond. More specifically, we can tell it O-glycosidic bond, because the bond is formed via the joining of the oxygen molecule. Okay, and because this galactose is in beta form, so it is called beta glycosidic bond. Okay, so if we see the maltose, there are two molecules of glucose which are joined via the O-glycosidic bond, and here, these molecules are similar, and in lactose, these molecules are dissimilar. So in the disaccharide molecule, the monosaccharide units can be either similar or dissimilar. And these are crystalline, water soluble, and sweet to taste. You can also see this is a sucrose molecule and this is a trehalose molecule. So the only difference between the maltose and trehalose is the alpha and beta unit. So can you tell me that which one is alpha and which one is beta?
Maltose alpha, trehalose beta.
Maltose alpha, trehalose beta.
Okay. And you can see this is sucrose molecule. One is glucose and one is fructose. And if you can remember that in the classification, we have learned that disaccharides can be further classified into two classes, like reducing sugar and non-reducing sugar. So in the reducing sugar, they contain free aldehyde or keto group. So if I can see in the anomeric carbon, it has a free oxygen, free hydroxyl group, or this is called the free aldehyde group. And if it also contains a free keto group, then it is also reducing sugar. So in that term, lactose, maltose, trehalose, all are reducing sugar. The maltose is called malt sugar because it is found in malt. Lactose is called milk sugar as it is found in milk. Then we come in the non-reducing sugar. This is sucrose, which doesn't have any kind of free aldehyde or keto group, because in the anomeric carbon, you can see the hydroxyl group is bound with CH2. Okay, and sucrose is also called table sugar. It is also known as cane sugar or beet sugar. Okay, trehalose is found in some bacteria, fungus, plants, and invertebrate animals. So we'll discuss about the glycosidic bond. The glycosidic bond is a covalent bond that joins two molecules of monosaccharide or two molecules of sugar to another group or carbohydrate itself. Okay, so the molecule can be either carbohydrate or any other molecule, because when you read about the nucleic acid, you will know that there are other glycosidic bonds where sugar molecule is not bound with a sugar molecule. Okay, so they are formed between the hemiacetal or hemiketal group of saccharides and the hydroxyl group of some compounds such as alcohol. So you can see this is the hemiacetal group of maltose, and it comes with the hydroxyl group of number 4 carbon of alpha D glucose. Okay, so it forms alpha D glycosidic bond, and in the lactose it forms beta D glycosidic bond. And in the sucrose, it formed actually alpha 1,2 glycosidic bond, because this is number 1 and this is number 2, 1 and 2. So it termed as alpha 1,2 glycosidic bond. So there are different types of glycosidic bonds like S, means sulfur, nitrogen, carbon, or oxygen. We'll discuss only about the oxygen or glycosidic bond in the carbohydrate part. Okay. If you have any question, just ask. So the disaccharide part is really so short, and oligosaccharide part is also short. Next we will come about the oligosaccharide part. We know that disaccharide is only 2 molecules of monosaccharide unit joined, and in the oligosaccharides there are 3 to 8 molecules of monosaccharides, but in some books or paper you will find also 3 to 6 molecules of monosaccharides in the glycoside or oligosaccharides. And likewise disaccharide, oligosaccharides, in the oligosaccharides the monosaccharides are held together by the glycosidic bond. For example, here you can see the raffinose. Raffinose contains galactose, glucose and fructose, and these are the glycosidic bonds. The oligosaccharides are found in many food, and not only the oligosaccharides, their derivatives also found in many foods, like genital oligosaccharides produced from pustulan. Pustulan is a molecule. This is also a carbohydrate molecule or glycan molecule, which is produced from lichen pustulatum, which is a fungus. You can also see this is a gluco-oligosaccharide produced from sucrose, human milk oligosaccharide, which is found in the breast milk. And there are also many other oligosaccharides and their derivatives, and it is mentioned here. You can read for later. Oligosaccharides are also often added to the commercial food as a sweeteners or fibers. So, if we want to know that if we eat oligosaccharide, what will happen? So normally oligosaccharides are indigestible, and human doesn't contain the enzyme that can break down the oligosaccharide. That means break down oligosaccharide into monosaccharide unit. So the enzymes which are required to break down the oligosacharide into 3 monosaccharide units are absent in the small intestine. However, there is one oligosaccharide which is called maltotriose. Maltotriose means maybe 3 glucose molecules. So other oligosaccharides generally reach in the large intestine, and they are actually fermented via the bacteria. In fact, they are beneficial bacteria which are present in the large intestine, and they are fermented and produce some form of energy. Okay. So if those large intestinal bacteria breaks the oligosaccharides in the large intestinal lining, they sometimes produce some beneficial nutrients which have some good effect on human health or other animals. Many oligosaccharides act as soluble fiber, which may help prevent constipation. So if we know about some fiber-like food, we eat some vegetables which are highly rich in fiber containing vegetables, then those are not actually digested properly inside this intestine. So those form some bulk and they prevent constipation. Okay, but sometimes if we eat a lot of those vegetables or fruits, then those may cause abdominal bloating or gas formation. So likewise, if oligosaccharides are ingested in a large amount, they can also produce abdominal bloating and excessive gas. As we know that oligosaccharides are broken down by the large intestinal bacteria. So sometimes they are also used as prebiotics. So prebiotics are non-digestible nutrients that selectively promote the growth of normal intestinal bacteria that may have beneficial effects on the large intestinal lining. So when these oligosaccharides are taken as a prebiotics, they actually have some effects on the bacteria which are present in the large intestine, and those are actually good bacteria for us, and they promote growth of the normal intestinal bacteria. Okay. So till now, if you have any question, you can ask. If you have any question, you can ask. No question? No question? So if you have, okay, we have a question, ma'am. It is very helpful for us if you talk into Bangla sometimes. Bangla bola jabe na যেহেতু তোমাদের সাথে ইন্টারন্যাশনাল স্টুডেন্ট আছে আর ইউনিভার্সিটির কনসেপ্ট আমি আগেও বলেছি ইউনিভার্সিটির কনসেপ্ট হচ্ছে ইংলিশে বলা ওকে তোমাদের যদি কোনো কোশ্চেন থাকে তাহলে তোমরা পরে এখানে আমাকে আস্ক করতে পারো যদি যেটা না বুঝো সেটা আমি হয়তো বাংলাতে আমি ট্রাই করব বুঝিয়ে দিতে ঠিক আছে Okay, if you have no question, I will proceed.
ম্যাম একটা কোশ্চেন ছিল
Sure, বলো।
অলিগোস্যাকারাইড অ্যাজ প্রিবায়োটিক্স এটা একটু আরেকবার বুঝিয়ে দেন।
Okay, prebiotics হচ্ছে ওইসব নন ডাইজেস্টিবল নিউট্রিয়েন্ট যারা লার্জ ইন্টেস্টাইনে থাকে এবং তারা অ্যাকচুয়ালি হচ্ছে লার্জ ইন্টেস্টাইনে আমাদের অনেক ধরনের ব্যাকটেরিয়া থাকে সেই ব্যাকটেরিয়াগুলো আইদার হার্মফুল অথবা হচ্ছে বেনিফিশিয়াল ওকে সো প্রিবায়োটিক্স এর কাজ হচ্ছে ওই বেনিফিশিয়াল যে ব্যাকটেরিয়া সেই বেনিফিশিয়াল ব্যাকটেরিয়ার গ্রোথ গুলাকে বাড়ানো যাতে কিছু কিছু খাবার যেগুলো আমাদের বডিতে ডাইজেস্ট হয় না সেই খাবারগুলোকে ডাইজেস্ট করতে সাহায্য করে এবং মোর যে নিউট্রিয়েন্ট সেগুলো আমাদেরকে সাপ্লাই দেয় এগুলো আসলে অনেকটা সিম্বায়োটিক রিলেশনশিপ মেনটেন করে সো প্রিবায়োটিক গুলো যখন খাওয়া হয় তখন তারা হচ্ছে লার্জ ইন্টেস্টাইনের যে নর্মাল ইন্টেস্টাইনাল ব্যাকটেরিয়া যেগুলো হচ্ছে বেনিফিশিয়াল ব্যাকটেরিয়া তাদের গ্রোথটাকে আরো বেশি বাড়ায় দেয় এবং আলটিমেটলি ওদের থেকে আমরা কিছু নিউট্রিয়েন্ট পাই Okay. Any more question? Okay. So you can go to the next slide. So we have finished monosaccharide in the last class. We have finished the disaccharide and oligosaccharides. Now we will start polysaccharides. So from the name poly, we can know that yes, there are many monosaccharide units. So usually there are more than 8 molecules of monosaccharide in the polysaccharides. And obviously they are the polymers of monosaccharides. Sometimes they are termed as glycans and their molecular weight is more than 20,000 Dalton. Okay. So, can you hear me? Hello?
Yes, ma'am.
Yes, ma'am.
So আমার হঠাৎ করে লোডশেডিং শুরু হয়ে গেছিল এই জন্য হচ্ছে আমি বের হয়ে আসছি আবার আচ্ছা আমি আবার স্টার্ট করতেছি Okay, so the molecular weight is more than 20,000 Dalton. For example, starch, glycogen, chitin. So you can see there are a lot of glucose molecules here in the starch, also in the glycogen molecule. And in the chitin, you can see that there are some variations at position 2 carbon. So this is called glucosamine, and this is N. That means N number of glucosamine molecules can join together to produce a chitin. Okay, so we'll next come to the classification of polysaccharide. Based on the structure, polysaccharide can be classified into 2 groups. One is the homopolysaccharide and one is the heteropolysaccharide. So in the homopolysaccharide, homo means the same types. So in the homopolysaccharide, if we break down this polysaccharide, we will get single molecules. That means same molecules of monosaccharides like starch, glycogen, cellulose, chitin. And they actually provide some structural functionality. For example, cellulose is involved in the cell wall formation in plant, because we know that plant cell has cell wall, but animal cell doesn't have any cell wall. So the cell wall is produced from the cellulose. And they can also serve as storage form and used as fuel. For example, glycogen and starch. Glycogen is stored in the liver of animals, and starch is stored in seeds or tuber like of food. And then comes the heteropolysaccharide. So heteropolysaccharides means these polysaccharides contains different types of monosaccharide units. In homopolysaccharide, we can find same monosaccharide unit, but in the heteropolysaccharide, the units are different, like hyaluronic acid, heparins, or chondroitin sulfate, keratin sulfate, etc. They actually provide extracellular support for organisms of all kingdom. For example, if you know about the bacterial cell, they have some layers on the bacterial cell, which are called peptidoglycan layer. We will discuss later about these things. And also, if you know about this extracellular space, there are some stuff like called matrix, which are involved in holding the cells together. Okay, and they actually provide protection, shape, and support the cells and tissue and organs. Okay, so both the homopolysaccharide and heteropolysaccharides can be either branched or unbranched. So if you can see in this picture that this is an unbranched homopolysaccharide, and all the units are same, so they are shown via the yellow color. And this is a branched homopolysaccharide, so we can see here is a branch. Okay, and the heteropolysaccharide, this is a unbranched or linear form of heteropolysaccharide, where you can see that there are two different monosaccharide units are present. And here in the branched heteropolysaccharide, you can see there are three different monosaccharide units are present, and this is the branch. So then we can know, we will learn about the classification, which is based on function. There are two types of polysaccharides based on function. One is the structural and one is storage. We have also already discussed about this thing, like structural polysaccharide contributes to the structure formation, like cellulose. Cellulose is present on the cell wall, and chitin is present on also the exoskeleton of some animals. We will discuss about this later. And storage, we have already discussed that starch is present in the seed of plants or tuber-like plants, and glycogen is present in the liver of animals. So we will discuss about some well-known homopolysaccharides and well-known heteropolysaccharides. At first, we'll discuss about the starch. So you can see this figure. This is potato, and these are some grains. So you can see this is rice, this is pulse, some corns are also present. I don't know how many seeds are here. So starch are the storage form of carbohydrate in plants. They are stored mainly in the tubers. You know that potato is very rich in carbohydrate source, so the potato contains a lot of starch. That means the whole potato is a starch, but there are some amounts of protein maybe present there. And seeds are the storehouse of starch. Okay, so starch is heavily hydrated, and they have many exposed hydroxyl groups available to hydrogen bond with water. So we will discuss briefly about the structure of starch in the next slide. And they contains the two types of glucose polymer. One is called amylose and one is called amylopectin. So what is in the amylose and amylopectin, we will discuss. You can see this picture. This is the figure of amylose. You can see this is a linear form of polysaccharide. So in this amylose, the monosaccharide unit is alpha D glucose. This is a long unbranched chain, and this alpha D glucose is joined to another alpha D glucose via alpha 1,4 bond. So the glycosidic linkage is actually written like here alpha 1, then there is a arrow sign, Then the next carbon of the next molecule is the 4, and then you can close the bracket, and this is bond. So in the amylose, if we break down the amylose, we will find a lot of alpha D glucose. So in the left side, this is the non-reducing end, so you cannot reduce here, but in the right side you can see this is the reducing end, and this is called amylose. But the amylopectin is a branched polymer, and the molecular weight of amylopectin is up to 200 million Dalton, and this is highly branched. So branching point is from 1 to 6. So the glycosidic bond is termed as alpha 1 to 6 glycosidic bond. You can see here, this is the number 1 anomeric carbon, and this is the number 6, that means the terminal carbon. So they are linked via the O-glycosidic bond. Okay, so if we just see an overall picture, this is the black color is the amylopectin, and the blue color is the amylose. Okay, so next we can go to the next slide.
ম্যাম আমার একটা কোশ্চেন ছিল।
Sure, ask me.
অ্যামাইলোপেকটিনে ব্রাঞ্চে হচ্ছে লিঙ্কটা 1 আর 6 এ হবে, বাট যেটা লিনিয়ার ফর্মে আছে সেক্ষেত্রে ম্যাম 1 আর 4 এ হবে না লিঙ্কটা?
Yes, this is 1 and 4.
অ্যামাইলোপেকটিনে
হ্যাঁ, অ্যামাইলোপেকটিনে ব্রাঞ্চ থাকে। সো তুমি যদি এই ফিগারটা দেখো তাহলে দেখবা যে অনেক জায়গায় এটা ব্রাঞ্চ থাকবে। সো অ্যামাইলোজ আর অ্যামাইলোপেকটিন কিন্তু একসাথেই থাকে। তুমি যদি এই পাশে যাও, এই ফার্স্টের ফিগারটা দেখো, এই যে আমি যদি এই পাশটা বলি অ্যামাইলোজ, তাহলে এই ব্রাঞ্চিং পার্টটা বলবো অ্যামাইলোপেকটিন, ঠিক আছে? সো স্টার্চে অ্যামাইলোজ আর অ্যামাইলোপেকটিন কিন্তু একসাথে মোটামুটি মিক্সড অবস্থায় থাকবে। ওকে, এনি মোর কোশ্চেন? So what do you think? What is the function of starch? What is the function of starch? What could be the function of starch? Starcher কাজ কি হতে পারে? আমরা তো starch খাই, we eat rice, we eat potatoes, right? Why do we eat rice or starch or potatoes, whatever you call? Why
produce energy?
Produce energy. So whenever we are lack energy, okay, we consume a lot of energy, then we should eat some carbohydrate containing food, and mainly we actually depend on rice or potatoes, right? So those are full of starch. So next we'll discuss about the glycogen. The structure is almost same. Glycogen and what is called glycogen and starch are same almost, but the branching is very compact in the glycogens. Okay, so in the glycogen we can see there are also alpha D glucose, and the joining is called alpha 1,4 glycosidic bond, and glucose are almost 50,000 subunits are present in the glucose, and the branching point is alpha 1 to 6. Okay, they are highly branched, and if we can see in the structure of the glycogen, then we can we know that we come to know that the branching points are found in every 8 to 12 residues, which is very compact, and this type of branching is not present in starch. So in the starch the branching is very light, but in the glycogen the branching is very compact. Okay, so the main storage polysaccharide in an animal cell is glycogen, which is found in the liver, and the weight is about 7% of the weight, and it might be also present in the skeletal muscle, but in very smaller amount. So each branch is glycogen end contains a non-reducing unit, and so if we count how many reducing unit and how many non-reducing units are present in glycogen, we can see that if the glycogen molecule contain n branches, then it has n plus 1 non-reducing ends, but only one reducing ends. So you have to find out which one is the reducing and how the reducing ends or non-reducing ends are calculated. Okay. So breakdown of glycogen starts from the non-reducing ends. So when we do not have adequate amount of glucose inside our body and we do not take a lot of food during our fasting time, then the storage form of glycogens are started to break down. Okay. And this process is called glycogenolysis. We will read about these things in the metabolism part, that in the glycogenolysis, the glycogen is broken down into glucose to serve the energy. Okay, so when the glycogen started to break down, then glycogen breakdown is started from the non-reducing part, but
Ma'am?
not, yes.
গ্লাইকোজেনের মানে পলিস্যাকারাইডটাকে ভাঙলে গ্লুকোজের সাথে আর কোন মলিকিউলটা পাওয়া যাবে?
শুধুই গ্লুকোজ, কারণ গ্লাইকোজেনে শুধুই গ্লুকোজ থাকে।
মানে গ্লুকোজের একটা হচ্ছে 1,4 লিংকেজ আর একটা 1,6 লিংকেজ, দুইটা।
হ্যাঁ, 1,4 লিংকেজ আর 1,6 লিংকেজ, তো লিংকেজ ভাঙবে হচ্ছে নন রিডিউসিং এন্ড থেকে, ঠিক আছে? নন রিডিউসিং এন্ড থেকে ভাঙবে, সো 1,4 লিংকেজটাই ফার্স্ট ভাঙবে এবং একটা করে গ্লুকোজ বের হয়ে আসবে। তোমরা ইন ডিটেইলস পড়বা, যখন গ্লাইকোজেনোলাইসিস পড়বা তখন তোমরা জানতে পারবা। কেমন? এনি মোর কোশ্চেন?
ম্যাম থ্যাংক ইউ। নো ম্যাম।
ওকে, সো we can ask that why, because in our blood we found that there is a carbohydrate which is called glucose. Okay, so we maybe all of us know about the diabetes, right? In the diabetes, the glucose is elevated in the blood. So we can see that glucose provides us the instant energy. So why the glycogen is stored in the liver instead of glucose? Okay. And here is the answer. Because in the liver, glycogen concentration is about 0.01 micromole. Okay, so this amount of glycogen is very less, and if we compare this amount of glycogen with the glucose concentration, this is about 0.4 mole of glucose. So if we see that 1 micromole equal to 1 into 10 inverse 6 mole, so we can easily compare that how many this amount of glucose is very high. So if this amount of glucose was stored inside the liver, then the size of the liver would be 6 times higher than the normal liver. Okay, and this amount of glycogen is not soluble in the water, and therefore it has no osmolarity effect on the cytosol, because the glycogen is stored in the hepatocytes. Hepatocytes mean the liver cells. So if this glycogen is remain insoluble, so they doesn't have any effect on the osmolarity. What is mean by osmolarity? What is mean by osmosis?
অবিশ্রবণ।
অভিস্রবণ, okay. What is the definition? Osmolarity definition কি? So osmolarity হচ্ছে যদি আমরা একটা semi permeable membrane রাখি, if there is a semi permeable membrane and on the one side the concentration of the solution is higher, and the another side of the membrane, if the concentration is of the solution is lower, then water will pass from the lower concentration of solution to the higher concentration of solution until the concentration of the both side of the membrane is similar. Okay, so যদি একটু বাংলায় বলি, আমাদের কি সাথে আজকে সব নেপালি স্টুডেন্ট আছে? Do you have any international student today?
Yes, ma'am.
Okay, so if we tell that if glycogen was soluble, so and those are stored inside the hepatocyte, then the concentration inside the hepatocyte would be higher. Okay, so the concentration will be thicker. Then in that case, the cells, the water molecule from the blood would be entered into the hepatocyte and causes the changes in the osmolarity. Okay. So now, if hepatocyte contains 0.4 mole of glucose instead of 0.01 micromole of glycogen, then the osmolarity would be elevated significantly, which I have already discussed, and that is why the water molecule would be inserted into the hepatocyte, and once the water molecules inside into the hepatocyte, the cell becomes swell, and ultimately this causes the lysis of the cell or rupture of the cell. Okay, so that is why glucose is not stored in its monomeric form, rather glycogen is stored inside the liver. This is not the only reason. There is another reason, like the external concentration of blood is about 5 micromole. So concentration in the blood is the mammal. This is the concentration. So this is, if this was 0.4 mole and this is 5 millimole, then the free energy change for glucose uptake into the cells against this very high concentration gradient would be very large. So there is a free energy change, because maybe all of us know about the thermodynamics law, like energy remain constant and energy can be transformed from one state to another state. So if this concentration of glucose was present inside the hepatocyte, then the free energy change would be always large. Okay. Any question?
Ma'am, can you repeat the last line you have said?
Okay, the free energy change for glucose uptake into cells against this very high concentration gradient would be very large. Okay, so when we eat rice, for example, okay, if we eat a lot of rice, then those rice are actually digested and metabolized. So digested into smaller pieces because we eat starch, and then those are broken down into glucose. Some glucose are instantly used, and the extra glucose are produced glycogen. Okay, so these extra glucose should be entered into the hepatocyte to produce the glycogen. So if those glucose were stored instead of glycogen, then this uptake of the glucose need more energy. Okay, that is what I have said here.
Thank you, ma'am.
Okay. So we have finished starch, we have finished glycogen. Now we'll discuss about another homopolysaccharide, which is dextran. You can see here is the teeth and here is the gum, and these are called, do you know what is called? These are called the dental plaque. Okay, I know that all of you have this dental plaque. So so the dextrans are called bacterial or yeast polysaccharides, and these are the polymers of alpha D glucose. You can see this is the structure, and the linkage is alpha 1 to 6. Okay, and there are also branches, like some dextran molecule might have branches on the alpha 1,3 branches most of cases, and some has alpha 1,2 branch, and some might have a branch at the alpha 1 to 4 link. Okay, so here in the dental plaque, there are some bacteria, because all of us have some bacteria inside our teeth. That is why we have to brush our teeth every day. So in this dental plaque, there are bacteria, and these bacteria actually stick to this dental plaque, and this stickness is responsible because of this dextran. Because of this dextran, bacteria can stick with this dental plaque. Okay, so the dental plaque is rich in dextran, and this is a source of glucose for this bacterial metabolism. Okay, so here in the dental plaque, the bacteria got a lot of energy from the dextran molecules, but not from the glucose molecules. Okay. Then come another homopolysaccharide, which is cellulose. We already have learned that cellulose is the structural homopolysaccharide, which is present on the cell wall of plant cells. So you can see this is the woody part of a plant. So all the woody part of the plant contains a large amount of cellulose. So this is the first abundant molecules in the nature. Okay, this is a tough, fibrous, and water insoluble molecule. So the cellulose molecule is linear. If you can see this structure, this is just two glucose molecules have been shown, but the branch has been open. So the cellulose molecule contain beta D glucose. So in the glycogen and in the starch, we found that there is alpha D glucose, and in the cellulose, the difference is it contains beta D glucose, and the cellulose might contain 10,000 to 15,000 beta D glucose in its structure. And ultimately, the bond is called the beta 1,4 glycosidic bond. Most of the animals cannot digest cellulose because they do not have the enzyme which have the ability to break down this beta glycosidic bond. Okay, but animals like cow and goats, they have the enzyme called cellulase, which can break down this beta glycosidic bond. Okay. Because human doesn't have this cellulase enzyme, they cannot fully digest the cellulose containing food or vegetables. Okay, for example, we eat various types of vegetables, like আমরা শাক খাই, তাই না? বিভিন্ন টাইপের শাক খাই, সেই শাকগুলা হচ্ছে ডাইজেস্ট হয় না। Okay, next come the chitin. Chitin is another homopolysaccharide, but if we can see the structure, this is the derivatives of monosaccharides. Okay, derivatives of monosaccharides. So here you can see this is the acetyl group and this is the N. So this monomeric unit is called N-acetyl glucosamine. Okay, because this is the glucose molecule, at the number 2 position there is an amine group or nitrogen group, and there's an acetyl group is linked here, so it is called N-acetyl glucosamine. So in the structure you can see this terminal carbon is in top and this terminal carbon is in down. Okay, so one is faced on the upward and one is faced on the downward. So the monosaccharide units are joined by the beta 1,4 glycosidic linkage. And you can see here, this is a gene beetle. You can find this beetle somewhere. The exoskeleton of these insects are made up of chitin. That is, it made a hard exoskeleton of many arthropods. And you can also find it in lobster, crabs. Okay, chitin is called the second most abundant polysaccharide in the nature. And this homopolysaccharide is also form fiber-like molecule, and they cannot also be digested by the vertebrates. Okay, so we have already finished the homopolysaccharide. So till now, if you have any question, let me know. If you have any question. No question? You have understood everything? Yes. Ask me. So in the in the class we have started from disaccharide. We discussed about the reducing and non-reducing disaccharide. We have learned about glycosidic bond. And then we come to the oligosaccharide. We learned that oligosaccharides are not digestible, and we come to know that they can be used as prebiotics. And we have learned about several oligosaccharides and their derivatives, which can be found in the food. Then we come the polysaccharide, and we come to know the classification of the polysaccharide, and we discussed about different homopolysaccharide like starch, glycogen, cellulose, and chitin, right? If you have no question, then we will start the heteropolysaccharide, okay? If you have any question? No question, okay. So heteropolysaccharides are sometimes called glycosaminoglycan, okay, or GAG. The GAG, or they are also termed as mucopolysaccharide. They are actually long linear polysaccharide, and they contain repeating units of disaccharides or double sugar. Like if you can, that means there are different molecules, different monosaccharide unit, because in the heteropolysaccharide, the monosaccharide units are not the same type but different. So in the mucopolysaccharide, they contain two types of monosaccharide units, but there are also several different types of monosaccharide units. So in the two monosaccharides, usually you can found either N-acetylglucosamine or N-acetylgalactosamine. So we have already learned about the N-acetylglucosamine. Okay, so if it is galactose, then it would be N-acetylgalactosamine. And we will found that there is a uronic acid. So that might be either D-glycuronic acid or L-iduronic acid. This iduronic acid is the C5 epimer of D-glycuronic acid. Okay. So some glycosaminoglycan might contain sulfur group, and which is that is esterified with the glucose or carbohydrate part, and GAGs are highly polar. They can attract water because they have the polar ability, and they can usually attract water. They can be used in the body as lubricant or shock absorber. We will discuss briefly in the next slides, and they can be found extensively in animals and bacteria, but they are largely absent in plants. However, some GAGs are found in the fungus also. Okay, so there are you can see the examples like chondroitin sulfate or dermatyl sulfate, heparin or heparan sulfate, carotene sulfate, hyaluronic acid. So we'll start with the hyaluronic acid. Hyaluronic acid, if you see this structure, this is a uronic acid of glucose. So this is glucuronic acid, and this is the N-acetylglucosamine. I have discussed already that what is N-acetylglucosamine. This is the nitrogen, this is the acetyl group, this is the N-acetylglucosamine. Okay, so the D-glycuronic acid is attached to the N-acetylglucosamine at the 3 position. Okay, the anomeric carbon is attached to the 3 position, so it formed a beta 1 to 3 glycosidic bond. However, if the next part, if the N-acetylglucosamine attached to the D-glycuronic acid, then the bond would be beta 1 to 4 glycosidic bond. Have you understood this one? Have you understood this one? So
No, ma'am, please repeat.
okay, so if if you can see this part, this one. So if we consider this one is the glucosamine, and this one is the N-acetyl, what does it called? So one is the glucuronic acid, and another is the N-acetyl glucosamine. Okay, so this one is glucuronic acid, and this is N-acetyl glucosamine. So the bond is beta 1 to 3, and then this glucosamine is bound to the glucuronic acid. This bond is called beta 1 to 4. Okay, so that is what I have told you. So the bond between glucuronic acid and N-acetylglucosamine, if I tell it NAG and AG, so glucuronic acid and NAG is bound with the beta 1,3 bond, And then after that one, there is another glycuronic acid. So NAG is bound to glycuronic acid via beta 1,4 linkage. Okay. So in the hyaluronic acid, there are around 50,000 repeats of disaccharides. So in total, we will find 1 lakh monosaccharide units in this hyaluronic acid. Okay. And because of these units, the molecular weight would be several million kilodalton. The hyaluronic acid is found, let's see in the next step, found mainly in the synovial fluid, okay, the vitreous humor. So in the synovial fluid is mainly found between the joint. So if we can see the elbow or knee, there are joints. So if this synovial fluid was absent between these joints, then they might be destroyed because of the agitation. Okay, ঘর্ষণের জন্য হাড়গুলো ক্ষয় হয়ে যেতে পারত. Okay, so this synovial fluid actually serves as the lubricant. Okay, lubricant, and this synovial fluid contain the hyaluronic acid. So hyaluronic acid actually give the mechanical support of the joint. Okay. And in the vertebrate eyes, there are vitreous humor. We know that here, sorry. So we know that in the vertebrate eyes, there is aqueous humor. We call it tear, and there is vitreous humor in the later part of the eyes. So in the vitreous humor is a gel-like substance, and this vitreous humor contain hyaluronic acid. And you can see this gel-like substance is thick and viscous and slightly sticky. Okay. It is also present in the cartilage, because there is also cartilage, that is the white part of the bone, and also present in the tendon. So this hyaluronic acid provides tensile strength and elasticity. There is an enzyme called hyaluronidase, which can have the ability to break down the hyaluronic acid, and it is found mainly in some pathogenic bacteria. Okay. Some animal species have this hyaluronidase enzyme, which actually break down the hyaluronic acid so that the sperm can enter into the ovum, and the glycosaminoglycan coat of the ovum can be hydrolyzed, and sperm can penetrate easily inside the ovum. Okay, so that they can fertilize. Then next we'll come to the chondroitin 4-sulfate. We have already discussed that the glycosaminoglycan are highly sulfated. That means some molecules are highly sulfated, sulfur containing molecule. So we can see the example that in the chondroitin 4-sulfate, we can see this is the glucuronic acid. The terminal carbon has been carboxylated, and this is the N-acetyl galactosamine 4-sulfate. Okay, so this is 1, 2, 3, 4. The number 4 carbon has been sulfated. The sulfur group is here, and this is the N-acetyl group. So these disaccharides are present here. The bond between glycuronic acid and N-acetyl galactosamine is beta 1 to 3, and likewise the heparin, the N-acetyl galactosamine 4-sulfate and glycuronic acid is contain the beta 1 to 4 glycosidic bond. Okay. আচ্ছা, আমি একটা ব্যাকগ্রাউন্ড সাউন্ড শুনতে পারতেছি আরিফ হোসেনের কাছ থেকে, তুমি কি কাইন্ডলি মিউট করবে? ওকে, দেন usually the chondroitin 4-sulfates are attached to some specific proteins, thus they can be termed as proteoglycan. We will discuss later about the proteoglycan. Okay, we have seen that the hyaluronic acid is found in the synovial fluid and cartilage, and chondroitin 4-sulfate is found mainly in the cartilage and bones. Okay, they are also found in the tendon and the ligament, and also in the valves of the heart. Okay, and also in the, all of the aorta. So the students of DVM will and also biotechnology, they will briefly discuss, they will learn about these things in their physiology class. And then there is another compound called dermatan sulfate or dermatan 4 sulfate. They are actually present on the skin, and they are responsible for the pliability or flexibility of the skin or elasticity of the skin. They are also present in the blood vessel and heart valve. The glycuronate is replaced by the iduronate here, which is the C5 epimer of glycuronate in the dermatan sulfate. Okay. Next, we will discuss about the carotene sulfate, which is another heteropolysaccharide. So you can see here, this is a galactose. Okay, there is nothing else. This is a galactose molecule. ওকে, আমি অলরেডি শ্রাবণী, আমি অলরেডি বলেছি যে আমি 1.5 ঘন্টা পড়াবো এবং একদিন পড়া বেশি হয়ে গেলেও আসলে এগুলো আমি তোমাকে যেহেতু ভিডিও দিয়ে দিচ্ছি, তোমরা এটা পড়া দেখে নিবা, কোনো সমস্যা হওয়ার কথা না। ওকে, so in the carotene sulfate there is galactose, and also there is a N-acetyl galactosamine 4 sulfate. So sorry, in this should be the 6 sulfate. This is maybe the structure is wrong. It should be 4 sulfate, so the sulfate should be here, and this is the N-acetyl galactosamine. Okay, so in the carotene 4 sulfate, the disaccharide units are about 25 molecules, and no uronic acid is present in the carotene sulfate. Okay, they are present in the cornea, in the bone, in the cartilage, and you can see here they can be found in the dead cells like horn. This is the hoof, this is hair, nails and claws. Okay, that means thaba. Thabar মধ্যে পাওয়া যায়, শিঙে পাওয়া যায়, তারপরে চুলে পাওয়া যায়, খুরে পাওয়া যায়। ওকে, কর্নিয়াতে, বোনে, কার্টিলেজে। Okay, then comes the heparin sulfate. So we have finished hyaluronic acid, then chondroitin 4 sulfate, keratin 4 sulfate, then comes the heparin sulfate. In the heparin or heparin sulfate, maybe you have learned about heparin, which function as a anticoagulant. That means they actually coagulate, they actually prevent the coagulation of blood. Okay, so you can see here, this is the iduronate 6 sulfate and iduronate 2 sulfate, and this is the N-sulfated glucosamine. So you can see here, this molecule is highly sulfated. There are 4 molecules of sulfate, 1, 2, 3, 4. So this is very rich in sulfate molecule, and the bond is alpha 1 to 4 glycosidic bond. This is mainly produced by the animal cells, and mainly by the mast cell, which is a type of leukocyte cell. Okay. Then we discussed earlier about the peptidoglycan. If you know that there are 2 types of bacteria, like gram-positive bacteria and gram-negative bacteria. So the bacterial, there are plasma membrane of the bacteria, and this part is called the cell wall of the bacteria. Okay, this cell wall is made up of peptidoglycan layer. In this peptidoglycan layer, there are some heteropolysaccharides, and this peptidoglycan layer is made up of, here you can see the structure, that N-acetyl glucosamine and N-acetylmuramic acid. So we can name it shortly like NAG and NAM. So these NAG and NAM are joined via alpha 1,4 glycosidic linkers, and this NAM is linked in parallel with some, this is a peptide cross bridge. Okay, this is called the peptide cross bridge, and they are highly cross-linked so that they can be protected from the antibiotics or any other things. Okay, say they form envelope, and they prevent cell swelling and lysis. Okay, so you can see that the gram-positive bacterial cell wall has one peptidoglycan layer, and the gram-negative bacterial cell wall has one peptidoglycan layer, but it has two lipid membrane or plasma membrane, and this peptidoglycan layer is located in between of the membrane. Okay, but here in the gram-positive bacteria, there is only one plasma membrane and there is a peptidoglycan layer. Okay. Then comes the glycoconjugates, which is the last part of this of our lecture. But we will discuss about 4 types of glycoconjugates. So glycoconjugates are nothing but the molecules where the carbohydrates are actually covalently liquid, protein, peptides, lipids, or saccharides. I mean other glucose or sugar molecule. Okay, so they are actually formed via a process which is called glycosylation. You will learn about different glycoconjugates like glycoprotein, proteoglycan, glycolipid, glycosphingolipid. So these glycoproteins are mainly proteins where small amount of glucose are bound. The proteoglycans are actually carbohydrate, but small amount of proteins are attached. Glycolipids are mainly lipid, but some amount of glucose are attached to the lipid, and glycosphingolipids are a subtype of glycolipids. And these glycoconjugates has different function, like they are involving cell-to-cell interactions, cell-cell recognition, cell matrix interaction, and detoxification process. So we'll discuss about the proteoglycan. You can see in the picture that the blue color is shown, which is a protein molecule, and this green color is the carbohydrate molecule, which can be a heteropolysaccharide, and these carbohydrate molecules are actually bound to the protein molecule via this covalent bond. So they are called proteoglycan. Okay, they are mainly found in the extracellular matrix. So they are present outside the cell wall. So one or more sulfated glycosaminoglycan chains are joined covalently to a membrane protein or a secreted protein. This proteoglycan are mainly. Hello, আমি একটা ব্যাকগ্রাউন্ড সাউন্ড পাচ্ছি, তোমরা কি সাউন্ডটা বন্ধ করবে? কেউ একজন সাউন্ড অন করে চলে গেছে, সাউন্ডটা একটু অফ করো। কাফি 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 তুমি সাউন্ড অফ করো। কাফি তুমি সাউন্ড মিউট করো। Okay, then GAG chain bind to the extracellular protein through electrostatic interaction between the protein and negatively charged sugar moieties on the proteoglycan. So they are actually bound to the we know that we have the cell membrane, and in the cell membrane there are different types of protein. So the proteins are actually present on the extracellular space or bound to the plasma membrane. So these molecules are actually bound to those protein via electrostatic interaction. Okay, and they are major components of all extracellular matrix. Then comes the glycoprotein. Sorry, this okay, maybe I have missed something. Okay. Right. Okay, I have missed that about this part is actually for proteoglycans. So glycoproteins are mainly proteins. They are also found in the extracellular matrix, and they are also involved in the interaction between cells. So they help to join two cell each other, so they can tightly held together. And sometimes glycoproteins are involved in recognition, cell to cell recognition. Okay, and then comes the glycolipids. So these are mainly lipids where carbohydrates are attached. Like this is the lipid membrane. Okay, the lipid bilayer membrane, you can see it in the plasma membrane. These are the sugar residues, and sugar residues are joined to the lipid surface. So these are called the glycolipids. They are mainly found in the cell surface of the cell membrane, and they are responsible for the maintenance of the stability of the cell membrane. And likewise, the glycoprotein, they are also involved in the cellular recognition. Like when some bacteria attack to our cell, then the bacterial components are um, bacterial components are recognized via these types of glycolipids. Okay, they are important for immunoresponse. Okay, like maybe you will in later learn about antibody and antigen interaction. So though here you will learn more about the glycolipids. Then comes and the subtypes of the glycolipids, which is mainly called glycosphingolipid. This is a sphingosine molecule. You can see that this is a large chain structure. Okay, so every zigzag contains one carbon molecule, and there is a double bond. This is the amino group, and this is a hydroxyl. Two hydroxyl groups are present. This is the smallest form of sphingosine. So these types of lipids actually called actually attached to this sphingosine molecule. Okay, and with this sphingosine, one carbohydrate molecule is attached, and these form the glycosphingolipids. They are also found in the plasma membrane. Like you can see, this is the lipid bilayer, and this is an inserted protein, and this is a glycosylated protein, and this glycosphingolipid is found in the outer surface of the protein. Okay, so this is the lipid molecule, and this is the carbohydrate part, and that is found on the outer extracellular surface of the plasma membrane. Okay, they contain they are mainly found in the neurons, and they are involved in the nerve conduction. So you some of you will learn about the action potential. Like if I punch you, or if someone just punch you, you will get some or someone just touch you, then you will get that sensation via the nerve conduction. Okay, so the nerve sensation will go to your brain, and then it come back toward that part. Like if you touch your hand, then you get the sensation on that hand. So this is called nerve conduction. And the glycosphingolipid is also involved in the myelin formation. So in the neuron cell, there are two parts. One is the I forgot the part. One is the cell body, and one is the extended part where you will get the myelin sheath, and this myelin sheath is rich in glycosphingolipid. Okay, they also play a role in the signal transduction in cells. So there are a lot of cells in our body, right? So the different cells maintain contact between them. So they maintain their contact, and they actually transfer signal between them. So this signal transduction is also maintained via this glycosphingolipid. Okay, there are different types of glycosphingolipid found in our body, like cerebrocyte, ganglioside, and globocytes. So that is the end of today's lecture. All of this lecture has been prepared from obviously Google and Leninger principle and biochemistry Satyanaran. Okay. So if you have any question till now, let me ask. Let me inform. No question? No question? Can you hear me?
Yes, ma'am.
Okay. Then ask me question if you have any problem.
No question, madam.
No question. Okay. So, DVM student, give your ID at first. Do not enter any other student's ID. Okay. At first, DVM student, give your ID in the chat box.